right, so welcome to the session, and as was just announced, I will be speaking about the detection and clinical interpretation of variants using Nanopore. And the main topic of my talk today is really the implementation of Nanopore sequencing for routine clinical diagnostics of rare diseases, which typically has like three components. You have the sample collection and wet lab part. That's not so much my field. Then you have the analytical part, where you have to find the variants, annotate them, and so on. And then you have the clinical interpretation using some clinical decision support system. I will focus today on the middle part here, which is the computational analytics of Nanopore data so that we can reliably call the causal variants. Why are we interested in that? Now, with the long-read technology in general, and specifically here with Oxford Nanopore, we have a lot of new analytical options. If you follow the traditional route via genome alignment and then consensus, what you gain is you have perfect haplotype phasing, so you can separate the reads by haplotype, and you get access to a lot of dark regions of the genome we hadn't have access before. But what we can also do now is genome de novo assembly, either globally, like in the telomere-to-telomere consortium, or just locally around an interesting complex regions with maybe a complex structure variants from an inversion, duplication, and so on combined. And again, genome de novo assembly gives us very nice haplotype phasing. And on top, we get the methylation, which is also phased by haplotype in the two alleles. So that's an add-on we just heard that we don't get otherwise. So that allows us actually to look now at very nice phased haplotypes instead of these ugly read alignments that you are used to from IGV. We can now basically just assemble two alleles for each patient. And we can now see within a family perfectly which allele was inherited by who, and how could that relate here, for instance, in a long insertion. How could that relate to the disease? Does it match the segregation of the disease, and so on? So that is a totally different view on a disease than read alignments. Now, to benefit from that, we obviously have to introduce it into the clinics, and there are, as you might know, still a lot of problems, technical problems and hurdles to overcome. So for that reason, we founded the LONGER consortium, which is Clinical Long Read Genome Initiative of Germany. That's why Long Germany. And it is supported by Oxford Nanopore. And our goal is here to have multiple medical centers, university medical centers that test the same technology, bring them in the routine. We are also open for other sites. If you think you are close to getting Nanopore into diagnostics, you can contact us. We want to use a clinically well-characterized cohort of more than 1,000 individuals. So we have 1,000 samples in the main study, and we already finished 80 pilot samples. And then we basically want to demonstrate the added diagnostic value. We want to show that it's actually sustainable, computationally, the IT infrastructure and so on. And we want to benchmark for accreditation of the laboratory-developed test. Good, streamlined pipelines and SOPs will hopefully be the result. So if you want to accredit the laboratory-developed test, you have to actually benchmark on many levels. As I said before, sample collection, here we need high molecular weight samples, all the wet lab preparation, then the IT infrastructure. For Nanopore, you need a lot of NVIDIA GPU servers because the base callers need that. And then you have all these NGS analysis software, which obviously is not yet broadly tested for clinical diagnostics. So that will be my main topic today, quality control for all the analysis. So in the pilot study I used for that, we had 2 genome in a bottle, GAP cell lines, that everyone is using for benchmarking, so as a reference data set for benchmarking, and then 68 real patient samples, all distributed across the 4 centers with the latest pore and chemistry. And we wanted to specifically choose cases that have complex stuff like repeat expansions, low complexity regions, pseudogene duplicates, pathogenic haplotypes like OPN1, and imprinting disorders with methylation. Here's the overview of the clinical samples from Tübingen. I only show that to give you the stats we have. For instance, on average, we do 35x coverage. So we prefer for diagnostics to have coverage between 35 to 40x. That is what we need. And then by that, we get in most samples close to 100% of the regions are covered by at least 20 reads. We had a relatively short N50 in these samples because we just took the same routine as for short reads, so we didn't specifically enrich for high molecular weight, but still about 14 kb. Oops, I jumped too fast. And the analysis on average took us about 26 hours. That might be interesting for you if you want to have rapid diagnostics. Here's the overview of the yield. We get most of the time around 120 to 130, which is exactly our 35x. If we get to 130, we get almost 40x, and at 100 gigabases, we get around 30x. The difference of these few samples down here is mostly that the pores close very quickly. Luckily, most of the pores had a good behavior, and we could top up one time. But in some cases, well, even top up doesn't help, and then you probably have to redo the sample. That still happens. So Genome in a Bottle, just as an introduction, this is a large project where the consortium, GAP consortium, has sequenced a few cell lines, 2 trios and 1 single index case extremely deeply with all technologies, and then they provide this high confidence benchmark set in the non-dark regions, so in the good to align regions with short reads, and then they have these difficult regions, difficult meaning hard to align for short reads because of repeats. And they were sequenced really with all technologies you can imagine, even some that I think don't exist anymore, like the linked reads. But that gives you a really high confidence variant call set, which you can use to benchmark. That's what we did. In Tübingen, we developed our own big nanopore pipeline, including all these read alignments, assembly, RNA, and so on. And unfortunately, I have the wrong link. We renamed it to MEC LR because we didn't want the company name in our tool name. So you have to replace that down here by MEC, Medical Genetics Long Read Pipeline. Okay, so very similar pipeline is also available from the GitHub of ONT. It's called WF Human Variation, and it uses almost the same tools. So it's quite homogeneous, actually. There's only a few really good tools that everyone is using, like Minimap, Deep Variant, Sniffles, and so on. First, we looked at SNV and indel precision and recall. So the triangles on the top, these are the SNVs, and the circles are the indels. And we used CLAIRE3 and the deep variant, which the old version is Pepper Margin deep variant. The new deep variant 1.5 is better, but just came out 2 weeks ago, so we couldn't use it yet. You can see that at 20x coverage, your SNFs are already perfect. They reach 99.9% accuracy. For indels, it's a different picture, and you might know that gap errors are one of the problems in Nanopore, and they benefit from high coverage. So the higher the coverage, the better they get. We reach a precision of 95 at 50x coverage. And there's a slight difference between the tools. Right now, CLAIRE3 works better than the old deep variant. We have to see now if the new deep variant closes the gap. And the recall is a similar picture. For SNFs at 20x, you have already perfect recall. For indels, we still gain recall when we go for deeper coverage. So to summarize that, SNFs are pretty much solved problem with Nanopore. For indels, we still have to go some way to improve them because we do not reach the perfect sensitivity we want. However, if you look for diagnostics, you are mostly interested in protein coding, and there we are significantly better, and the reason is simply that in intergenic regions you have much more homopolymers than in coding regions. And I said, as before, you have to go deep. With 40x, you already reach, for the indels, a pretty good sensitivity and precision on coding, and for SNFs, well, you only need 20x. But that's one of the reasons we go for these 40x or 50x coverage to make the indels better. Still, the question is, can we further improve indels? I mean, we would like to have them also at 99% and not at 94%. And for that, we have now the duplex reads, which are a recent introduction from Nanopore, where basically not just one strand is sequenced, but the second strand is captured and then sequenced right afterwards. And that's kind of a random chance, it seems like. So right now, up to 30% of the reads will be duplex reads. It depends a bit on how much you overload your flow cell. The more DNA you put in, the less duplex you get relatively, because they fight against each other. I guess the second strand versus a new read coming in. So that's screenshots directly from the Nanopore website, and you can see here the old chemistry, the latest chemistry with simplex reads, and then the alignment with the duplex reads, which is much better. So that gives us hope, but we wanted to test it ourselves, obviously. And we plotted the quality over the length of the reads. And what we can see with the simplex reads, so where just one was sequenced, that you have a bit less than 30 Q, but at around 20K, the quality drops off, something you probably also know from short reads. But if you take the duplex reads, now you sequence first forward and then the reverse trend. So you have from both sides, you have good quality, one time on forward, one time on reverse. And you suddenly see that the quality stays high up to the end of the reads. We had very few reads more than 35 KB. That's why it's getting fuzzy in the back there, because it's a small data number problem. So that's one good thing. And it also results in more reads having at least Q20 and significantly more reads having at least Q30. And that then we could analyze by aligning the reads from these reference samples HG001 and HG002. This one is also known as NA12878, if that name is more familiar to you. And we can see that at simplex reads, we get an error rate of 2%, and with duplex, it's a quarter of that, 0.5%. And that's a combination of the mismatches, insertions, and deletions. And we see that for insertions and deletions, we get about 5 times better with the duplex reads. And also interesting, because of this effect I just explained, that reads stay high quality to the very end, you have much less soft clipping. So for simplex reads, at some point, Minimap soft clips because the quality gets bad. But for the duplex reads, only 12% are soft clipped, so 90% less, basically. So the most important for us is here, with 5 times less gap errors, we hope that we can improve the indel diagnostics, but we couldn't do it yet because of the problem that we don't get enough duplex reads from a single flow cell. In the next step, we will pool across all the 4 centers. We have 8 flow cells of HG001, and with that, we will get the at least 30x duplex coverage and can then report, hopefully in a later talk, if that worked out for us. Structure variance is another thing that is really good with Nanopore. So here we have an example with Nanopore compared with BioNano and short reads. And we tested that as well using 2 tools, Sniffles and QtSV. And as you can see, Sniffles is basically good even with 5x coverage. They already reach 90% precision and recall, and they get slightly better with more coverage. QtSV, on the other hand, is pretty bad at low coverage, so it has very low recall, but it seems to be tuned for high coverage. So for high coverage, QtSV outperforms Sniffles and reaches almost 95% recall. We assume if we select for high molecular rate long reads, we might even get the 95% or better recall. So as a conclusion, I would suggest using Sniffles most of the time, but if you have more than 40x coverage, look at QtSV. It's quite good. And you can see that with Sniffles, there is no gain anymore after 30x and only a slight gain from 20x. But with QtSV, we saw an advantage of high coverage. Now, for the final few minutes of my talk, I just want to show you a few examples of these pilot samples, these 68 pilot samples we did for clinical diagnostics. In general, you have advantages with long reads for the compound heterozygotes because you don't need trios anymore. You can phase a single child and can see if it's a real compound or if the variants sit on the same allele. I already showed you structure variants, which we now can find with 95% recall and precision. We haven't really tested mobile elements, but I guess that's similar, and inversions. But I want to focus a bit more on these types: repeat expansions, methylation imprinting, and duplicate genes. I found them a bit more interesting. We made a little tool. It's extremely simple. It sorts for repeat expansions, it sorts the reads spanning the repeat expansions in the shorter and the longer allele, and then it plots their length. It's not yet enabled for motif changes and so on, so this is really an early work in progress, but I just wanted to let you know how we analyze the data. And with that, we can then look into specific cases. Here is Huntington's disease case, the gene HTT, and it has a 53 copies of a CAG repeat. You can see here sorted the longer and the shorter allele. And that basically solves the case very easy. You can even see it by eye. You don't actually need a program for that. Here's another one. In that case, both alleles are a bit longer than the reference. This is an ataxia in SCAR3. But you can still easily sort the two alleles into their two lengths, and then likely having the longer one, the 77 CAG repeat, being causal for this ataxia version 3. And with that, normally you have a diagnosis. Then we have more complicated repeat expansions, and that typically happens when they are very long. Then you have problems to visualize them nicely in IGV. Instead, what you get are these short purple bars here that indicate a large insertion. And specifically in that case, again, the shorter allele was already longer than the reference. It had like 30 repeat copies, like 90 bases long of a GAA repeat in FGF14, and then allele 2 has more than 300 repeat copies, and you can see that here it falls into these two length areas. What we do here also in orange, you see the reads that fully span the repeat, while in green you see, and green and purple, you see the read that only manage to go into the repeat but not fully cross it, and from the other side. And especially the ones that don't fully cross, they often lead to a slight misinterpretation of the length. So if you have enough coverage, like 40x, I would suggest only to use reads that fully span the repeat expansion to estimate the length. Even then, it's a simple repeat. Nanopore has some problems with them. The lengths will never be exact on a single motif copy, but they will be close. And in these cases where you have such an increase in repeats, it's not a problem if you predict 301 or 302. It doesn't really matter. With the duplex reads, again, I hope to get even more tight results here on the length distribution. Good, and then you have the cases. Maybe I want to mention here this FGF14 is very interesting. It was just released this year that FGF14 is a causal repeat, deep intronic in ataxia, and I talked to colleagues who say that possibly up to 30% of the unsolved cases in their biobank can be solved by that one. So I find that really interesting that we still find new repeats that nobody has had on their map before. Okay, here in that case, it's a very long hexanucleotide repeat in the C9orf72 gene in ALS disease. And as you can see here, this one has like almost 12 kb insertion. And with that, because we had on average only 15 or 14 kb and 50, we have problems to span that, and we see a big drop in coverage. So the shorter allele is well covered, and this allele is not as well covered, and we have some clipped off reads which just didn't make it over the repeat. So the longer your repeat, obviously the longer you want your reads to be, and the more coverage you need. We can still see it, but here I wouldn't trust the diagnostics. I would probably add either more coverage or a secondary method to validate it. Also interesting is, because of the haplotype phasing, we can sometimes see events like that. Here in a SCAR4 ataxia, we had the expanded allele and the normal allele in two different patients, And the expanded allele always comes with a pretty crazy haplotype that is full of SNPs. So many, many more SNPs than you see in the other haplotypes that is not expanded. I'm not sure if there's some evolutionary biology behind that, if selection doesn't work anymore, or if that gene is just dead now. I don't want to claim that just based on 2 patients, but I found it extremely interesting to see these 2 separate haplotypes that are so different from each other. Now, the other topic is that we can get DNA methylation right out of the sequencing, and we can phase it. So we can separate the methylation into the 2 haplotypes as well, because we have sequence, variants, and methylation. And here a very nice tool, MethylArtist, plots that and can also show these so-called imprinting loci, where from one parent you inherit a methylated and from the other parent a non-methylated allele, for instance. And that can lead to diseases, for instance, in uniparental disomy, where you accidentally have 2 copies of one parent and then suddenly they are both silenced or something like that. So that makes it extremely interesting. We can look at these loci. Here is one case in autism spectrum disorder, gene SNRPN. And you can see here in a control that you have intermediate methylation, red and blue. And then in the case you have only, I think blue is only non-methylation, or only methylation. I think it's weird because IGV swaps that depending on how you load the samples, the colors. I think it's highly methylated. And when you look at that specific case, it's the FMR1 gene, and it's a methylation that affects a repeat expansion. So there is a repeat expansion here in the FMR1 gene. It's on the X chromosome in male, so it's a homozygous, basically homozygous expansion. And if it's longer than 200 copies, the locus is silenced by methylation. At least that's as far as I'm, as a bioinformatician, understand the whole thing here. So here you see the methylation of that locus, much more methylation here than here. And here you see the repeat expansion. So at the same time you see here in color the methylation level, red versus blue, and in purple here you see the inversion, the repeat expansion, not inversion. So as I'm not a geneticist myself or medical doctor, I always go to Google the problem and find a good paper, and here this one explained me what is happening, that in that syndrome in most FXS patients the FMR1 gene is epigenetically inactivated following the expansions over 200 triplets of CGG. So that's what you see there, expansion plus methylation change, all in one go. All right, here is a final example, and because of time that's my last one, for the duplicate genes. You are probably all familiar with the SMN1 and its pseudogene SMN2, which also can have multiple copies SMN2, but SMN2 is not generating a fully functional protein, or at least only, I think 15% of the SMN2 proteins are functional, the rest is not. So if you lose SMN1, like here, by this deletion, you will not have enough protein, although depending on the number of copies of SMN2, you get some protein. And that creates the disease spinal muscular atrophy. And as you can see here, we have 3 controls and then the gene itself, and in that case we simply had a large deletion of that gene. With that, I want to finish. I have shown you that we can benchmark using these GUP cell lines how well our nanopore implementation for the clinical diagnostic works. We can later use that for accreditation or IVDR and so on to prove that we are consistently keeping up the same benchmark values. We can do that for SNPs, indels, and SVs. I have also shown you that indels are our main remaining construction site where we have to improve. And we have shown that we get exactly what we expect for patients. We can solve complex repeat expansions, duplicate genes, and so on. And here you see our collaborators from the 4 university clinics in Germany, Aachen, Berlin, Hannover, and Tübingen. Big thanks again to Oxford Nanopore for supporting that project with flow cells and also technical support. And additionally now also NVIDIA joined us to give us IT technical support and compute power on GPU servers, which we will need for the amount of patients we will do. And here was from the kickoff meeting just 2 months ago, and some people from my lab who couldn't be there and also contributed a lot. Thanks.